So what happened, say in mitosis, okay, there were five phases: prophase, prometaphase, then metaphase, anaphase, and telophase. This happens in mitotic cycle before the mitosis. During the interphase, we'll talk about G2 interphase. For example, here it is. Yeah, you can see the centrosomes form there. Okay, centriole pair. The chromosomes are not duplicated. They are duplicated but not condensed. Okay, this is plasma membrane, nuclear envelope, nucleus, nucleolus. Okay, this is the final stage before mitosis. Then what happened was centromes into centrioles. Then they produce microtubules. Then what happened? Cell division happens. Okay, chromosomes and the DNA replication took place in S phase, and during interphase they were uncondensed. Okay, you can see but a better picture for this. Okay, after that, what happened in the prophase? The first phase of cell division. In the prophase, what happens is the chromatin fibers. Okay, they started to become more coiled. Okay, they're getting more condensed, and then you can see the nucleoli or nucleolus disappeared. Okay, what happened here in centrosomes? They form a radial array. Okay, this you can see outside it. These are called as esters. These have a special role from to extend microtubules. Okay, these are called microtubules. Okay, now at this stage you can see the mitotic spindle is forming. Also at this point we can see the chromosomes with light microscope. Okay, you also see that like these centrosomes are like moving apart from each other at different location. And also what happened is, see, when the chromosome is duplicated, okay, these appears as two sister chromatids. Okay, at this point they are joined by the with centromere with the help of sister chromatid cohesion. Later on, what happened is during the prometaphase, the second phase, the more condensing of chromosomes occur, the nuclear envelope fragments, it discontinues. Okay, you can also see the microtubules extending from the each centrosome are joining. Okay, here the chromosome has become more condensed. You can see the microtubules joining together without touching a chromosome. Okay, but they are called null kinetochore microtubules because they don't have a kinetochore. For example, in this way, you can see the centromere. A centromere, two specialized protein formed at both ends of the sister chromatid, joined by the microtubules, known as, what do we call them? We call them kinetochore microtubules. Okay, so there are two kinetochore for one chromosome. One means two chromatid. One kinetochore is attached on one chromatid. Second kinetochore is attached on second chromatid. Where they are properly joined with what centromere. Okay, you can see this is the centromere. So sorry, centrosome. This is centromere. This is centrosome. They are extending the microtubules towards the kinetochore. The kinetochore is attached on centromere. On each centromere, on each chromatid, there is one kinetochore. So there are two kinetochore per one sister chromatid. So next phase was about metaphase. There are opposite poles. Okay, centrosomes are at opposite pole. Metaphasic phase what form. Okay, the equidistant, they are equidistant from each other, which means they are at equal distance from each other. They are at opposite side right now. Okay, the spindle is formed correctly now. Then the comes our anaphase, the shortest phase of the mitosis, a few minutes phase. Okay, what happens is you can see the chromosomes, the chromatid becomes the new chromosome. They are moving at a speed of 1 micrometer per minute. Now question arises, why like they are not formed, why like they are like not pulled like this way and like pulled like this way? Why it happens? Because of this the special reason, because the kinetochore is attached here, the centromere. Okay, they are pulled from this side. As they are pulled from this side, this formation occur like this one. This is the correct formation occur because at this point when you pull something from the middle, okay, the gravity is going there. They form this way as a V shape. You can say V or U shape. The what happened also? The cell elongates, means it enlarges, and this is his final point before it started to separate. Okay, by the end of anaphase, you can see the cell have equivalent and complete collection of the chromosomes. So now telophase started. At the time of telophase, what happened was two dot nuclei. You can see here they are forming. Okay, nuclei is reappeared. Spingle microtubules are disappeared. Okay, spindle is disappeared. The mitosis is complete. You can see cleavage furrow. The cell begins to divide from the middle. Centromeres, centrosomes are at new centrosomes are forming. Okay. Lastly, what happened? Cytokinesis. Cell proper divides two into daughter cells. Both nuclei form. Okay. Nuclear envelope form. Chromosome form. Mitosis is done. You can see in the meta picture, cell here breaks into two, division of cytoplasm occurs, two but daughter cells. Yeah. So some key terms we learn today is mitotic spindle. First of all, see what is a mitotic spindle. Okay, suppose this is a cell. Okay, it's a cell. What are these called? These are called centrosomes. Okay, one here also, and these rays which are emerging from it called ester. Okay, they form thread-like structure toward each other. What are these called? Microtubules. Okay, so for a zoom in microtubule, let me say zoom microtubules. Okay, for example, see this is a cylinder. Okay, and inside of it, it is hollow, means it doesn't have anything in it. It's hollow. They are tiny. It is made up of some fibers, tiny hollow fibers, which are further made up of a protein. We call it as tubulin. So now what happens in the cell? When the microtubules grow, okay, or they shrink, growing and shrinking, see if the microtubules grow, we can say elongate. It is done by adding tubulin proteins. Tubulin proteins. Adding more tubulin proteins. It means adding, which we call it process is polymerization. Polymerization. Okay. Now what happens if they shrink? If they shrink or get shortened, by losing tubulin subunits, or we can say proteins. We call it depolymerization. Now moving on, like the spindle microtubules begin to form at the centrosome in animal cells respectively. We can also say this as microtubule organizing center. Organizing center. Okay. So now each centrosome, for example, this is centrosome. It has a pair of centrioles. For example, this one is centrosome. Oh wait, uh, we can say this whole thing is centrosome. Okay, and one of it we can call it centriole. Okay, so the important part here is the centrioles are not essential for mitosis. Even they are destroyed, the spindle still forms. You can see in the plant cells they don't have centrioles. Plant cells don't have centrioles, but still the mitosis, the mitotic spindle form. How it is formed? It is formed by the centrosome material. Centrosome material helps in organizing microtubules. So what happened in the tail is, we can say before mitosis SBM, we can say in interphase, what happened was single centrosome duplicates, by which they stay near the nucleus, when the mitosis started, you can say during prophase and prometaphase. What happened? Centrosomes began to move apart from each other. Then also the spindle microtubules grow outward from them, which means like they grow from them. After that, like when the prometaphase ends, the each centrosome reaches at the opposite end. You can say here, like at North Pole, or you can say South Pole, just for explaining. Okay, a star-like structure. You can say this one. It is formed at both ends. These are short microtubules. Okay, and this star-like structure is known as ester. It formed around each centrosome. Then together centrosomes, spindle microtubules plus the ester made the mitotic spindle. Let's talk about the kinetochore role. So what happened is, for example, Okay, this is one chromatid, this is second chromatid. Okay, together they constitute a chromosome. Together this is known as chromosome. Okay, this is one chromatid, second one here. Okay, now at what happened is, now at this point I have marked in the red. Here centromere is located, a point where these two chromatids are attached to each towards each other. At there, a special type of molecule, we can say, it is formed on the both chromatids. We termed it as kinetochore. It is formed of proteins. Okay. Now, what happened during prometaphase? So, during the prometaphase, what just happens is, like some of these spindle fibers attached to the kinetochore, we call it as kinetochore microtubules. It is varied. For example, like in yeast, only one type of only one microtubule got attached to it. Okay, but in humans, several types of microtubule or several number of microtubules attached to the kinetochore. We can say in human it is 40. Okay, now what happens is when a microtubules, okay, wait, suppose this is a chromosome. Okay, this type of with this type kind of attaches here also. So microtubules are pulling the chromosome toward this way. This chromatid is pulled toward this way, this chromatid pulled toward this way. Tug of war is happening. Okay, now what just happens, it will break apart. But I'm of metaphase, all chromosomes are lined up here, you can see. Okay, we can say an imaginary line, we can call it metaphasic plate. Now we can see like spindle, it is completely formed. Okay, moreover we can see the non uh, kinetochore microtubules. You can see this one in this microtubule, they are connected but they have not have a kinetochore. Okay, they are connected with each other but they don't have a kinetochore. They are overlapping with each other towards the poles. Okay, they help in stabilizing the shape of the spindle. Moreover, we can see the esters. These esters, they are also connected to the cell membrane. Okay, they are help in the anchoring of spindle, like they'll provide the shape of the cell division. During anaphase, what just happens is, for example, these two are, this one is a chromosome. Okay, so what happens is like these two sister chromatids are holding together with a protein called at cohesin. What just happened? It is cut by an enzyme. We can call it as separase, which help in separating the sister chromatids. Sister chromatid separates here, separated by separase enzyme. Then the chromosomes we can say that then the each chromatid, I'm writing it CM. Each chromatid will form new chromosome or individual chromosome. These chromosomes move towards opposite end of the cell. Chromosomes, new chromosomes move towards opposite end of the cell, or we can say opposite poles of the cell. Now let's talk about how does they move. Now the how does chromosome move? First is we can talk about is Pac-Man movement. So we know that like the chromosome moves when the kinetochore is attached to it with the help of the microtubules. Okay, they pull it towards their each end, and then the chromosome moves. So what happened basically is here motor proteins on the kinetochores act like a small motor. Okay, what does happen there? Like they walk along the microtubule toward the pole. For example, we can see like this is a chromatid here. We can say new chromosome. Okay, you can say this is a microtubule which is attached to kinetochore here. Okay, slowly by slowly they are eating it. For example, and also this comes it closer. Now understand it is a way like this is a pole. Okay, what just happens here? This is a microtubule here. Okay, it attaches to this chromatid. Now what happened is we can think of like usually this chromatid is eating the microtubule. Okay, what happening is breakdown of microtubules, or we can say depolymerization. Like we can say slowly by slowly it is eating the microtubule and reaching toward the pole. Okay, now the second mechanism was reeling in, which means like here microtubules don't shorten at kinetochore, but it gets shorter at spindle pole. For example, see uh, this is centrosome. Okay, so what happened is they are like reeling the rope. For example, have you like if someone like fly the kite, what we do like we pull the string toward ourselves to pull the string toward ourselves. Okay, so during that time this thing is like this. For example, a microtubule is present here first. Now it start pulling, it come closer by closer. Okay, So now what happened is there are motor proteins attached to at the spindle forms, which helps to reel the chromosome toward them, and later on the microtubules also break down here. Depolarization, depoly depolymerization happens there also. I have a better picture. Wait. So now better understanding we can say at here you can see at the first method, Pacman method, what just happened here at the kinetochore, they are cutting the microtubules and slowly reaching there. But in the reeling in method, what happens? The spindle pole, it is also cutting the microtubules, but reaching at the opposite side towards the spindle. Suppose this it is in a vertically upward vertical position, right? Now this is vertically upward. Okay, you can see there here the gravities, these are coming toward this side. Okay, and here is opposite side, this is opposite, from there it is coming this side. So what about the other microtubules, like the non-kinetochore ones? What just happened, like they also help in stretching the cell longer. In anaphase, special motor proteins attach to these overlapping microtubules, making them walk them apart, like pushing the poles away from each other. Okay, this is done by ATP energy, or we can say chemically. At the same time, also, like these microtubules get go longer and longer by adding more tubulin units. Okay, later on at the time of anaphase and the ending or starting of telophase, you know that like the group of chromosomes, okay, forms at each end of the cell. Okay, it's telophase. Okay, and the cell is now longer than before. It is long, it is longer in length. Okay, the nuclear membrane are forming also. Then cytokinesis happens. The spindle deassembles. The spindle deassembles. Microtubules depolymerize and they recycle the tubulin for the future use.